Okay, welcome back everyone. So today we're going to see DNA replication. Okay, DNA replication. So we have seen the structure of DNA, we have seen the types of DNA, also we have seen the experiment to prove the DNA is a genetic material. Then we have seen RNA, all the types of RNA we have seen, and we have compared RNA in DNA. Now we need to see the replication, DNA replication. Okay, then we will see transcription, then we will see translation. All right, so what is replication? Replication means making copy of its own. Okay, when cell divides. So go back to your first year chapter, cell cycle and cell division chapter. When cell divides into two daughter nuclei, so the DNA is there inside the cell, right? So in short, let me show you. So we have this cell. Inside the cell we have DNA, that means inside the nucleus. We are not going to talk about this in detail. So there we have DNA. Now when cell divides into two daughter nuclei, okay, when the cell divides into two daughter nuclei, the DNA which was there inside this cell, the same DNA, they replicate. Okay, and the equal amount of DNA they distribute to those two daughter nuclei. So here we need to see how this DNA actually replicates. Replicates means what? Making copy of their own, like a Xerox copy. Okay, so when cell divides into two daughter nuclei, DNA also divides. Okay, so that equal amount of DNA should get those two cells. Okay, to do so they replicate. So there is a process. We're going to see this in DNA replication. We call it as a DNA replication. Okay, so in which phase of cell cycle the DNA replicates? The first year chapter, cell cycle. In which cell cycle? The first one is interphase. Do you remember or not? The interphase is divided into G1, S and G2. Then come M phase. M phase is prophase, metaphase, anaphase and telophase. DNA replicates at S phase. Okay, at S phase. So first year chapter, you need to go back, flashback to your first year chapter. So here we will see how DNA replicates. So for the replication of DNA, various enzymes are required. Okay, enzymes plays a very important role for the replication of DNA. So we need to see that enzyme. There are many enzymes which plays a very important role, but we are going to talk about the important enzymes only. So we're going to see one by one all the enzymes. Okay. So first we're going to talk about the various enzymes which help in the replication process. So we'll come to one by one along with their function. The first enzyme we can talk about Helicase enzyme. There are many enzymes, but we will discuss only the important enzymes. Okay, so Helicase enzymes are there. What they do? Helicase enzyme. First I'll write and I'll explain. Wait. So Helicase enzyme, it opens up the two helix of DNA by breaking hydrogen bond. Okay, it opens up helix by breaking two hydrogen bond. See, our DNA structure is a double helical structure, right? We have seen like this. Though here it seems like they are crossing each other, but they do not cross with each other, okay? They are spirally coiled with each other. So try to imagine the spiral stair. Spiral stair, you might have seen spiral stair. So just like that, the DNA strand, the 2 strand. So in my diagram, it looks like a cross because I cannot show you 3D diagram in this board. So try to imagine. These 2 helical structures are connected with the nitrogenous base pair, and the nitrogenous base pair, they make bond between each other. Purine pairs with pyrimidine, adenine pairs with thymine by making double bond, cytosine pairs with guanine by making triple bond, right? We have seen this. That bonds are actually a hydrogen bond, weak bond. It can be breakable easily. Okay, so for the replication to occur, the first of all, these 2 strand has to open up, so that new bond gets synthesized inside the strand. Okay, suppose, let me try to show you. So here my finger, this is 2 strand, okay? So they are like coiling like structure. Our DNA strand are coil like structure like this, right? They are connected, these 2 strands are connected with nitrogen base pairs. So for the replication to occur between those 2 strands, these 2 helix, my finger here is a helix, consider, okay? These 2 helix has to get open like that, so that here the new strand synthesis in both the strand. Here the new strand will synthesis, here again the new strand will synthesis. So this strand has to get open first, okay? So to open this strand, enzymes plays a very important role. The first enzyme is known as helicase enzyme. See, from entrance point of view, and any competitive exam, when you appeared in your future related to biology subject, in a PhD or civil service or medical exam, this DNA replication is very important, and the enzymes names are very important. Along with the rule, you need to remember. Okay, so be careful, pay attention, and try to clear your concept. So first, this 2 strand has to get open, so that the new strand synthesizes in both the 2 strand. Okay, then only they will make the copy of their own, right? They can transfer each copy in both this cell in equal amount of DNA. So these 2 strand has to open. So suppose from this point the replication is going to take place from this point. So what happened? The helicase enzyme, suppose this is a helicase enzyme. Helicase enzyme comes and bind here. Okay, helicase enzyme comes and bind here. Now when this helicase enzyme bind this strand to this strand, the helicase enzyme breaks, they start to break the hydrogen bond, because hydrogen bond helps in connection of those 2 strands, right? So they breaks the hydrogen bond, leading to the opening of the 2 strands separately. Okay, leading to the opening of 2 strands separately. So after breaking of hydrogen bond, the DNA strand, now the DNA strand will get separate like this. Okay, they will separate like this. Those 2 strands has to open, only then the new strand will synthesize. That means the new DNA strand will synthesize. Okay, so this enzyme, when they moves along those strands, they start to opening up all those strands. Try to imagine, try to bring images in your mind. Okay, you have to be a philosopher. If you are science student, first you have to be a philosopher. Think in your mind. Okay, visualize the images in your mind, then you write it down. So this enzyme moves along this strand. When they moves along this strand, it start breaking all the hydrogen bond which was attached, right? The hydrogen bond which connects the 2 nitrogenous base pair. It breaks the hydrogen bond. When they moves, it breaks the hydrogen bond and start opening to helix and so on. Okay, this is the first enzyme, helix. What they do, they open up the two helix of DNA by breaking hydrogen bond. So this is his role. This is his role. Okay, okay, topoisomerase. So next enzyme is topoisomerase. Now what helicase did, helicase opens up the DNA strand. Okay, this was our strand. It can be that direction also, that direction also. So now according to our concern, I am showing it a diagram like this. So here again, don't get confused. I can show like that also, I can show like that also, I can show like that also. So don't get confused in here. Okay, this is nothing. So here the helicase enzyme opens up to helix. The next topoisomerase, what they do, see, after opening up these two strands together, there is still a chance that those two strands, they may go back to their original position. Okay, because they always remain in helical strand, helical structure. They are always in a helix form. So that was their original position. When they open to helix, it remains tension in those two strands. The tension still occurs in those two strands because they are always in a helical structure. So when they open, suddenly it remains a tension in those two strands. They will try to go back to their original position. They will tend to go back to their original position because they always remain like this, right? So when they open up suddenly after breaking hydrogen bond, there is a tension. They tend to go back to their original position. For example, spring. Take an example for a spring. If we stress the spring, if we stress any spring, you know, when we stress, it becomes straight for some time. When we open our finger, then you will still go back to their original position, right? Just like that, because they have a tension, because they always remain in that position. So when we stretch it, it becomes straight for some time, and we release our hand, it goes back to their original position, right? So after helicase opening up those two strands, in those two strands, tension still remains. Okay, the tension still remains. So there is a chance it goes back like this in their original position. So to remove that tension, the topoisomerase help. This is another enzyme, topoisomerase. Okay, topoisomerase, what they do to remove that tension, topoisomerase cuts the strand. Okay, they cuts the strand. Try to understand. They cuts the strand and rotate the strand. While rotating the strand, it releases the tension. It cuts the strand and rotates it and reseals it. Okay, what topoisomerase do? Topoisomerase releases the tension of those two strands. How? By cutting the strand, rotates the strand and reseal it. Okay, the reseal is done by another enzyme again. So we will see. So first of all, topoisomerase, it releases the tension of two strands by cutting the strand and rotates it and reseal it. Okay, it releases the tension of two strands of DNA by cutting the strands and rotates it and reseals it and rotates it and reseal it. Okay, that is how topoisomerase do. So there is a tension, right? These two strands has a tension. They may go back to their original position. So to prevent this thing, the topoisomerase, they cut this strand, okay, They cut the strand of DNA, they rotates it, releases the tension, and then reseal it. You have to imagine the images in your mind only, or else you can see YouTube, 3D structure. If it will help you, try to see 3D structure of DNA replication. Okay, it cuts the strand, rotates the strand, and reseal it. Okay, so the reseal of this strand is done by another enzyme known as ligase enzyme. Ligase. The resealing of those two strands after cutting the topoisomerase, the reseal is done by ligase enzyme. It helps in joining of two strands after topoisomerase. Okay, so far what we have seen, the first is helicase enzyme. What helicase enzyme do? Helicase enzyme breaks the hydrogen bond of those two DNA strands and opens up the helix so that the replication can take place in both the strands, so that the new DNA strand can form in both the strands. Okay, so that opens the helicase enzyme. But after opening of those two helix, those two strands, there is still a tension that those two strands tends to go back to their original position. To remove that tension, topoisomerase, what they do, topoisomerase enzyme, it cuts these two strands, rotates the strand, releases the tension, and reseal again, reseal back. And this resealing of DNA strand is done by ligase enzyme. Ligase. Okay, so next we have 1, 2, 3, number 4. Next enzyme we have primase enzyme. Primase enzyme. What is primase? Primase, it is an enzyme only. It helps in the addition of primer. I'll tell you what is primer. It helps in addition of primer. What is primer? Okay, it has been resealed. Okay, here in my diagram, it has been resealed. So one strand has 3 prime. The end of that strand is going to have 5 prime here, but we will write the 5 prime here. Okay, the strand which is open, so we are going to put a 5 prime here. The 5 prime will be here only, but the opening part, okay, the point where it is the opening of the strand, we will put it here, 5 prime. Other strand going to have 5 prime, then 3rd prime here, right? So we know. So primer is a short segment of RNA. It can be DNA also. Wait, I'll tell you. It is a short segment of RNA. So for the DNA replication, RNA strain is required. So we call it as a primer in case of DNA replication. Okay, primer is a short segment of RNA, consists of 6 to 8 nucleotide base pairs. So it consists of 6 to 8 nucleotide sequence long. Primer is a short segment of RNA, consists of 8 length, it's about 8 to 6 nucleotide sequence long. Okay, RNA, the primer, that is primer, is a DNA dependent RNA molecule. Okay, one more thing, it is a DNA dependent RNA molecule. A RNA is a short segment primer is a short segment of RNA. It consists of 6 to 8 nucleotide sequence. It is edited on a DNA strand in both the strand. Okay, the primer is a piece of RNA, short segment of RNA, like that. It consists of 6 to 8 nucleotide sequence long. So it is edited in both the strand. Okay, it is edited in both the strand. So this addition of primer is done by an enzyme primase. Okay, primase. We will see that when we discuss the actual process of DNA replication. What primer do? That is the RNA. What RNA primer do? We will see that. But here we are going to talk about only the enzyme. So for now, remember, our primer is a short segment of RNA, consists of 6 to 8 nucleotide sequence, and it is a DNA dependent RNA molecule. That means it requires a DNA strand to perform its function. Okay, so just see, the primer can be DNA also, RNA also. Okay, the primer, it can be a short segment of DNA also and a short segment of RNA also. But for the replication process in DNA, it uses RNA primer, not DNA primer. Okay, that's you remember. So after primase, which help in the addition of primer, then it comes DNA Polymerase One Enzyme. So DNA Polymerase One Enzyme. Say RNA, that is the primer here. It's a short segment of RNA only, primer. So in DNA replication, the primer helps in addition of, in joining of other nucleotides. We will see that. But it is a RNA, and we do not want, or our body do not want RNA with DNA. Okay, because RNA is an unstable molecule. And it is not a genetic material, that's why, because it is unstable. So we do not need, or we will not take RNA, okay, along with the DNA. The RNA primer, its role is to aid some nucleotides. Once RNA primer aids some nucleotide, when their role is over, they need to taken out from the DNA strand. Okay, once the role of RNA is done, the RNA primer has to taken out from the DNA strand, and this removal of RNA primer is done by DNA polymerase 1. Okay, it helps in removal of primer. Now when I talk about primer, that means a short segment of RNA. It helps in the removal of primer and helps in fillings of gaps. Okay, so let me show you. So primer is a short segment of RNA, right? So primer, what they do, they aid in the addition of other nucleotide. Once their role is over, this RNA primer has to taken out from DNA strand, because it is a RNA and our cell will not take RNA as a genetic material. Okay, so they have to taken out. So RNA comes here and bind with the strand. Suppose this is an RNA segment. Now why in this, so in both the strand the primer binds. Okay, first this strand. Now why primer comes and bind here, why not there? Okay, why here in this strand, why not at the bottom of this strand? Remember, whenever or wherever there is a 3', and when we talk about 3', there is always a OH group free. That means there is always a hydroxyl group free. That is the functional group. It has a function to bind other molecules. Okay, that's why the RNA primer will going to bind with the 3' of OH group free with its 5' and 3rd'. Okay, remember, the primer is always going to bind the 3rd prime of the DNA strand of its 5th prime, okay, of its 5th prime. That's it. And the new nucleotide that is synthesis is always towards the direction 5th prime to 3rd prime. Please remember, when the new nucleotide gets synthesized, the direction is always from 5th prime towards 3rd prime. So 5th prime towards 3rd prime. So this RNA primer, we're going to add new nitrogenous base pair or new nucleotide sequences. Why the primer is required? Because primer it acts like a glue, okay, sticky, it acts as a glue, okay. For example, have you ever painted your house with color? If you did, you might know that before applying the colors on wall, we add primer, right? We add primer before applying any color. Why it is required? Primer helps, okay, it acts as a claim. Primer helps in holding the color as well as the wall along with it. So exactly the role of this primer is that it helps in holding of DNA strand along with the other nucleotide sequences. Okay, when the new nucleotide sequences comes and bind, it will bind from this RNA primer towards the 5th prime of DNA strand. Okay, towards the 5th prime of DNA strand. Now remember, the new strand or new nucleotide sequences always get synthesized from 5th prime towards 3rd prime. So 5th prime, this is 5th prime, this is 3rd prime. So it is going to get synthesized. Okay, I hope you understood. So this is the primer that has been added in this strand. Now in this strand, at which point the primer is going to come and bind? Here or here? Here we have 5th prime, here we have 3rd prime. Where the primer is going to come and bind? Again, when there is a 3 prime, always it contains a functional group, OH group free. Whenever there is a OH group free, the OH group is always going to bind any other molecule of their required molecule. Okay, so in this strand the primer is not going to come and bind here, because in this strand there is a 5th prime free. 5th prime always binds with the phosphate group, okay, 5th prime. So here we do not find OH group free, but here we have 3 prime here. So when there will be a 3 prime, there is always a functional group. So primer is going to come and bind it here in this strand, this of its 5th prime and 3rd prime. Now new nucleotide sequences always get synthesized from 5th prime towards 3rd prime. So 5th prime, that is 3rd prime, it is going to get synthesized. New nucleotide sequences is going to get synthesized here. That means the new strand is going to get synthesized. Alright, so this is the process. Again we will discuss when we discuss the process of replication. So that is the role of DNA polymerase 1. DNA polymerase 1, once the role of primer is done, because it is a RNA segment, so we do not want RNA to transfer from generation to generation, because it is not our genetic material. We want only DNA. So to do that, the DNA polymerase 1, okay, there is a DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. So here DNA polymerase 1 enzyme helps in removal of primer only. So they will remove primer, DNA polymerase 1. The DNA polymerase 1 comes and bind here. It is enzyme is what? Enzyme is nothing but a protein only. Suppose this is enzyme. So DNA polymerase 1 comes and bind here, and they are going to remove the primer only. And once the primer gets removed, the gaps is going to fill with another nucleotide. Okay, that is the role of DNA polymerase 1. I hope you are getting my point. Alright, so next, the very important enzyme. All these enzymes are helping in the process. Now the next coming enzyme now, it is the actual enzyme that is going to help in the process of replication. What is the number? Number helicase, topoisomerase, primase, ligase, DNA polymerase 1, 6, number 6. DNA polymerase 3, that is very important enzyme. This is the actual enzyme which is going to help in the replication process. DNA polymerase enzyme is a large enzyme, big enzyme. It has many subunits with it. Okay, DNA polymerase 3 enzyme is a very huge enzyme. It has many subunits. The first subunit is known as alpha subunit. Now each subunit has its specific role to play in DNA replication. Alpha subunit, what they do, they help in the addition of they help in the formation of bond between nucleotide. Okay, it helps in formation of bond between nucleotides. Okay, see, remember, when we are going to discuss DNA replication, we have to keep in mind, we should already know the function of each enzyme. When we discuss the replication process of DNA, we should already know all the function of enzyme. Then only you will understand. So before going to the replication process of DNA, first of all you memorize all the function of this enzyme. It will make you too easy for you understand. Okay, so here we have primers. Okay, so new nucleotide sequences are coming and bind with each other. So this bond between those two nucleotides, this bond is done. The addition of bond or formation of bond is done with the help of DNA polymerase 3 alpha subunit. Okay, so it's its role. Next subunit is beta 2 subunit. Now beta 2 subunit is the DNA polymerase 3 enzyme only. It is a subunit. Beta 2 subunit, it acts as a clamp again. It acts as a clip to hold the DNA polymerase 3 enzyme along with the nucleotide base pair. Okay, suppose this is DNA polymerase 3 enzyme. This beta 2 subunit, it acts as a clip. Suppose this is clip or something. Okay, it acts as a clip to hold this enzyme along with these two strands, the nucleotide sequences, so that they play their role. Otherwise, there is a chance that the DNA polymerase 3 enzyme might fall off. Okay, see replication, how it takes place. Suppose this is a strand. Okay, this is a DNA suppose this is a DNA strand here. This is enzyme come and bind it here. Okay, this is a strand where the replication is going to take place. The strand will be there only. The strand do not move. The enzyme is going to move. From this point, when enzyme come and bind at this point, they are going to synthesize all the nucleotide base pair. The new nucleotide base pair they are going to synthesize. Okay, once this point is done, synthesis of nucleotide sequences is done, they are going to move to other, they will move further. So here they are going to move like this. Okay, so when this enzyme, suppose this my hand is enzyme and this is DNA strand. This enzyme is holding this strand. The holding of this enzyme is done with the help of beta 2 subunit. The beta 2 subunit is going to come and bind between those two. Suppose this is beta 2 and going to come and bind here. Okay, it's going to act as a clip, so it holds the enzyme as well as this strand, so that the DNA polymerase enzyme do not fall off from this strand. Okay, so strand will be there, strand do not move, the enzyme is going to move. So from this point they will synthesize new nucleotide base pair or new nucleotide sequences. Now once it is done this point, they will move further come into the middle. Here they will again synthesis, and once it is done, they're going to come in here. They will synthesis new nucleotide sequences. Done work, so they're going to false off. So that is the process, how it works. Okay, so beta 2 subunit, it acts as a claim to hold the enzyme along with DNA strand. Okay, so last but not the least, we have one more enzyme, epsilon naught subunit. Epsilon naught subunit. DNA polymerase 3 is a large enzyme, huge enzyme, big enzyme, so it contains many subunits along with it. Each subunit has a specific role. So last we have epsilon naught subunit. What they do? Epsilon subunit, it helps in proofread. What does that mean? It helps in proofread. Now these new nitrogenous base pairs are adding, okay? They are synthesizing a new nitrogenous or nucleotide base pair. But there is a chance, okay, there is a chance the mistakes can occur. Out of 100%, there is a chance, 1%. Okay, 1% chance that when they added new nitrogenous base pair, they might also add wrong nitrogenous base pair, which lead to the mutation again. If they added any wrong nitrogenous base pair, it is going to result into mutation. So out of 100%, there is a 1% chance that they may add wrong nitrogenous base pair. To check that, epsilon naught subunit helps. Okay, once they added all the nitrogenous base pair, all the they synthesis new strain, the epsilon naught subunit is going to come and bind here. So they're going to check the new strain. They're going to check the new strain. If they find, if this epsilon subunit find any wrong nitrogenous base pair, they're going to replace that nitrogenous base pair with the correct ones. Okay, it has a very important role. So suppose this is a new strain has been synthesized. Okay, so epsilon naught subunit is going to come and bind here again. Here is, we have new nitrogenous base pair. That means new strain, we have new DNA strain. The nitrogenous base epsilon naught subunit is come and bind to the new strain. It is going to check one by one. They will move like that. They will move. They will move. If they do not find any wrong nitrogenous base pair, they will just fall off simply. But suppose from this point, correct nitrogenous base pair all okay. When they move to further direction, when they found, if they find any wrong nitrogenous base pair at the middle of the strain, they're going to replace that wrong nitrogenous base pair with the correct ones. That is his role. And there is a chance, few chances, okay? Out of 100%, 1% mistakes can occur. So epsilon naught subunit, it helps in proofread. What is proofread? If there is any wrong nitrogenous base pair has been synthesized in a new strain, it is going to replace it with the correct one. It is its role. Okay, so that's it about DNA polymerase 2. DNA polymerase 3, not 2. Okay, one more we have enzyme. One more enzyme we have. Enzyme number 8, I guess. Phosphorylase enzyme. Phosphorylase, it helps in the addition of phosphate. It helps in the addition of phosphate. Okay, this is enzyme. We will see it. So this is all the important enzyme which take part in the DNA replication processes. Why replication? During the cell division, from one single cell, it divides into 2 daughter nuclei. So we have known this. It divides into 2 daughter nuclei, right? During cell division, during gamete formation, in which phase? S phase of interphase cell cycle. First, the cell goes into G1, where the cell prepare everything to enter into the next phase, that is S phase. I don't know whether you remember or not. I know you have forgotten already, but still. So after G1, they will enter into the S phase. Now here at the S phase, this G1, G2, S and G2 are what? Their interface cell cycle. After G1, they will enter into the S phase, right? At S phase, the DNA replication occurs. Very important entrance point of view. Always repeatedly ask this question. In which phase of the cell cycle DNA replication occurs? So at the S phase, the DNA replication occurs, where the cell divides into two daughter nuclei. The cell along with the DNA, they also give the equal amount of DNA into those two daughter nuclei, so that they get equal amount of DNA. So the process that takes place, the replication, exactly it is we're going to talk about here, how it is going to take place. Okay, though in this diagram it looks so simple, but the actual process, it happens like that. For this to occur, various enzyme is going to takes part in the replication process. Okay, various enzyme is going to take part. So we have seen enzyme. First enzyme is what? Helicase enzyme. What they do? Helicase enzyme, for the replication to occur, these two strain has to get open, because they are in a coil structure. These two strain has to get open. Okay, they are in this form, this coiling like structure. Let me again show you. So they are in this form. Okay, they are in this form, but when new strain needs to synthesize, these two strain has to get open, so that the new strain is going to get synthesized. Okay, so this is done by helicase enzyme. How they do? They breaks the hydrogen bond. Okay, they breaks the hydrogen bond, this bond. They breaks only hydrogen bond. Okay, then the strain gets separated from each other. Now, but there is a tension, because they might go back to their original position, because they always remain in a helix form. Suddenly when they get open, they will tend to go back into their original position. So to remove that tension, another enzyme helps, topoisomerase. Topoisomerase, what they do? They cut those two strain, rotates the strain, releases the tension, and then rejoin it, reseal it. And this reseal of those two strain are done with the help of another enzyme. We call it as a ligase enzyme. Ligase enzyme always joins the strain. Okay, so after that there is one more enzyme. I left this one. See, after ligase enzyme, you add one more enzyme. Okay, single strain. See, I left this one. Single strain stabilizing protein. So there is one more enzyme. So after ligase enzyme, there is one more enzyme. You add this one. Okay, single strain stabilizing protein. So after ligase enzyme, single strain stabilizing protein, also it releases the tension, or it stabilizes the strain. Let me say stabilize. It stabilizes the strain. Okay, so after rejoined with the help of DNA ligase, the another enzyme called single strain stabilizing protein, they stabilize those two strain. Okay, they stabilize those two strain. Then after that, the primase enzyme act on it. What primase enzyme do? Primase enzyme, they help in the addition of primer. What is primer? Primer is a short segment of RNA. It is about 6 to 8 nucleotide sequences long. What the primer do? Primer helps, it acts as a glue. It holds the DNA strand as well as it helps in the addition of new nitrogenous base pair or new nucleotide sequences. I have given the example of primer which we used to paint in our house before we apply some color on it, right? Then to remove the primer, there is another enzyme called DNA Polymerase One Enzyme, which help in the removal of primer and help in the fillings of gap. Because primer is a RNA strand and we do not want RNA strand to pass from generation to generation. Once the role of primer is done, the primer gets removed with the help of DNA Polymerase 1. After that, the very important enzyme, it comes into play called DNA Polymerase 3. DNA Polymerase 3 is a very big and large enzyme. It has many subunits. The first one is the alpha subunit. Alpha subunit help in the addition of bond between two nucleotides. Then it comes beta 2 subunit. It acts as a sliding clamp. It acts the DNA polymerase enzyme along with the new nitrogenous base pair and also the DNA strand. Then come epsilon naught subunit. Epsilon naught subunit, what they do, it helps in the proofread. If there is any wrong nucleotide has been added in a new strand, it is get replaced with the correct one. So this is all about the rules played by certain enzymes. So next we're going to talk about the actual process of DNA replication in the next video.